Hello everyone, this is lecture 8 of course PE 500. In this lecture, we are going to cover the definition of nonlinear equations and their rules in bioengineering. So what is the difference between a linear differential equation and nonlinear differential equation? And what makes an equation nonlinear? Simply, we mentioned in the previous lectures that a linear function or a linear differential equation doesn't contain any derivatives or any powers. And if any of those exist in the differential equation, it converts it from a linear to nonlinear. So we can consider a nonlinear differential equation if it contains one of the following. The dependent variable or its derivatives are raised to power other than 1. For example, y squared or dy by dt raised to the power of 2. Also, if the dependent variable or the derivatives are products. For example, when you multiply y by dy dt or y by the second derivative of dy dt. Also, if you put the dependent variable inside transcendental functions such as sine, cosine, exponential, or log. So if there is any manipulation between the dependent variable and or the derivative, then it's considered as a nonlinear. And these nonlinearities prevent the use of superposition and often make analytical solution impossible or difficult to use. And some nonlinear equations cannot be solved analytically and may be solved numerically or graphically, as we are going to see. So as definition, a linear differential equation can be found on this form: dy dt plus a function of t or a constant multiplied by y equals to a function of t. But a nonlinear differential equation can be on this form: dy dt plus a function of t multiplied by a function of y. It can be y squared or sine y or cosine y equal to a function of t, where this function doesn't equal y, because if it's equal y, it will be converted to the linear version. These are some examples of the nonlinear differential equations. For the first, logistic growth, because p is multiplied by p, so we have p squared, and this is one of the things that we mentioned. If we found it, we will consider that the differential equation is nonlinear. The second is also c squared divided by c squared. The third, we have v cubic, so all of them are raised to powers other than 1, so we consider them as nonlinear differential equations. So if you made a quick comparison between linear and the nonlinear differential equations, according to the derivatives, in the linear we have only first power, but in nonlinear it may be multiplied or raised to powers, and according to the dependent variable, it appears linearly in the linear differential equation, but it can be squared, cubed, or within transcendental function in the nonlinear differential equation. For the superposition, it applies here and doesn't apply here. And for the analytical solution, it often exists in the linear differential equation, but it rarely exists in the nonlinear. And the behavior over time, it's predictable in the linear, but in the nonlinear, it can be chaotic or oscillatory, as sometimes a very small input to the nonlinear differential equation, it can lead to chaotic behavior or oscillatory behavior in the environment. So you don't have the ability to predict the output. So why do we study nonlinear systems or differential equations in bioengineering or in our field? Simply because most of the real-world bioengineering systems may involve nonlinear behaviors, which make these equations essential for modeling realistic biological processes. And these nonlinearities may arise due to some reasons. For example, threshold-based activation, and this happens when you find that the system has no effect until a given threshold, and it will start again to work. Or you can find saturation effects, where you have a system that increases by time, and until it reaches a certain maximum value or a specific limit, and you cannot increase after it, whether you're increasing or keeping the input. Feedback mechanisms, where you have a loop in your system to get a feedback from the output to the input to update it again, or a population interactions, such as the dynamics between the immune cells, cancer cells, and there are many more. So by including these points to our system, this will convert it to nonlinear differential equation or nonlinear system, which will require special treatment and dealing with them. One of the examples for the nonlinear systems in bioengineering is called gene regulatory network or GRNs. And GRNs describe how genes interact to control each other's expression. And we can assume that this network consists of a set of components collected together, and the output is the whole output of the system. So let's assume that each part of the network contribute to the output. So the final output will be the collection of these outputs, but including the interactions between the different elements or components of this network. But not all of the components will behave the same or will behave in a linear fashion, but one or more of these components may behave in a nonlinear dynamics. And this can happen because of multiple reasons. One of these reasons is like a threshold. Maybe a certain component is not active until it gets a specific value or a threshold, and then it will be activated. Or we have an activated system, and at a specific value, it will be disactivated. Also, we can have feedback loops, whether they are positive or negative in our system or network. Or we have saturation effects. For example, we have a component in this network where the output increases until it reaches a certain value, and at high protein concentrations, there are no more output. It will be kept at this level. So by including all of these in a big network or in our network, which is the GRN, then the final output or the output of this network isn't predictable, and it will be for sure nonlinear. One of the models which is common is called Hill equation, which models the rate of change of the gene product, G of t. And as you check this equation, you will find that we have different coefficients, and we have here a symbol called n. And n here reflects the Hill coefficient, and this reflects how sharply the gene responds. And in here can be 0, 1, 2, and so on. Beta is the maximum production rate. Gamma is a degradation or dilution rate. K is half maximal effective concentration or the activation threshold. And as we mentioned, G is the concentration of the gene product. This equation or this model introduced two things. Saturation, which we mentioned, it's like increasing the output, but until a certain threshold or a maximum capacity, the output will be the same, whether you are increasing or keeping the input. And the switch-like behavior, it's like toggling a switch between off and on states. So we can assume it like a threshold. We have an off state, and at a certain value, it will be on state, and so on. And combining both of them alongside the different nonlinearities in the network or the system, we cannot guarantee we have a linear model. We have now a nonlinear model. And depending on the value of n here, we will get the response of G of t. So by varying this value, we may get a flattened shape or an S shape or sigmoid response, and is controlled by this factor, which is called a sigmoidal response. And this factor is called degradation or dilution of gene product over time. Combining both of them together would make our system exhibit three things: saturation, switch-like behavior, and we describe both of them, and bistability. And the meaning of bistability is that we have two stable points. Let's assume it's stable in the off state and it's stable on the on state. So when you find a bistable system, then it's stable on two stable states depending on the initial conditions. So it's worth noting again that the initial condition is essential because it can make the system stable or unstable. So let's assume we have here 2 points, here and here, to understand the meaning of bistability. So as these 2 points, let's assume the system would be stable depending on the initial condition. So if we started the initial condition right here, then we can reach this point. And if it changes our initial conditions right here, we will reach this point. So depending on the given initial condition, you will reach a specific stable or non-stable point. And if you have two stable states in your system, you can call it a bistable system. And the Hill equation or the Hill model can be used as toggle switch in synthetic biology to create something called memory-like circuits in the cells. This is another example of neural action potential modeling. And here neurons accept complex firing patterns. And this neural firing follows the nonlinear dynamics. It's because some neurons stay at rest until a threshold stimulus is reached, and fire action potentials, when activated and return again to the rest state or enter oscillatory models under sustained input. So we have different phases for the neuron firing, and this behavior arises from the voltage-gated ion channels and feedback mechanisms if we are looking for the anatomy point of view. And we can model these using the Huxley model, using these two equations, where V here is the membrane potential, W is a recovery variable, I is external stimulation, and epsilon here, which is below 1, is the time scale separation between fast voltage and slow recovery, and others are factors. So if you look carefully at V cubic here, you will find that we have a cubic differential equation or a cubic nonlinear differential equation. So we can say we have here a cubic nonlinearity, and this will mimic the sharp threshold for firing. We have also minus W plus I, which is input current and recovery mechanisms, and dW by dt is slower negative feedback that controls refractory period. Using the Huxley model or these two equations, we have a system that accepts a resting state, and here we have a stable equilibrium spiking because of the limit cycle oscillation, and we can observe chaotic behavior under a strong external input or noise. And this model can be used in different applications, such as studying neural coding and signal propagation. For now, we have mentioned two examples, and we don't need to go in depth through each of them. We mentioned them to know that we have systems and real-world applications that are nonlinear, and we may find in your experiment that your system behaves nonlinearly, and we need to understand how to deal with these systems. So there are challenges. One of these challenges is the analytical solutions for this solution for these systems rarely exist or may not exist. So in most cases, nonlinear ordinary differential equations cannot be solved analytically compared to the linear ones. So the question is now, if we cannot solve these systems analytically, how do we understand these systems? For example, we have here a nonlinear system, and you may wonder, it's a very simple equation. It's dy by dt equals y squared plus t. So why cannot we solve this analytically? Or why there is no closed form solution exist for this equation or this ordinary differential equation? And the major problem now is, if these equations cannot be solved analytically, so how do we understand these systems and deal with them? And there's a big challenge. So although if you cannot use formulas and pen and paper to solve the equations, we can use something else. One of those is called numerical solvers, such as Euler methods and RK4. Also, we can use simulation and qualitative analysis. And two common techniques for the qualitative analysis is called phase plane analysis, and especially they are used for 2D analysis, and bifurcation diagrams. So the plan now, we have a system. We will check if it's linear or nonlinear. If it's linear, now we know the different ways to solve it, whether you're using analytical or numerical methods. But if it's nonlinear, then you will ignore analytical solution and you will stick with numerical or qualitative ways to solve it or understand the system. We discussed how to deal with the numerical methods such as Euler and Runge or RK4, and we are going to cover now the qualitative ways to understand the system. So in this lecture, we are going to cover the qualitative ways to understand the system, especially the nonlinear system. So we will start by the bifurcation diagram. So what is a bifurcation? So simply, a bifurcation occurs when small changes in the system parameters lead to qualitative changes in its behavior, such as appearance, change in stability, or the appearance of chaos or oscillation. So to understand this, let's assume that a patient got a therapy for a cancer, and this cancer is suppressed and the patient, and then at a critical point or a change of a certain value, the tumor increased again or the tumor spread again. So this critical point that converted the system or the patient from the stable state to unstable state, it's called bifurcation. So we need to understand at what critical point or at what certain value of a specific parameter or a set of parameters the system will convert to chaos or oscillation or become unstable and so on. So by studying the bifurcation and understanding the qualitative behavior or it changes visually, we can understand many things, such as how neurons start firing, why ecosystems collapse, when tumors regrow after treatment, or thresholds in genetic activation. So the idea now is that we have a certain parameter and we need to understand by changing that parameter what is the behavior of the system. So we can understand at what point we can call it a critical point or a set of critical points the system will behave differently, such as switching from stable to unstable. So by using the bifurcation diagrams, which are graphical representations, we can study how the behavior of a dynamical system changes as one or more of its parameters are varied. Again, this is important in the analysis of nonlinear systems, where smaller changes in the parameters can lead to significant qualitative changes in the system dynamics, such as appearance, stability, or chaotic behavior. So how do we plot it? Simply, on the x-axis, which is the horizontal axis, we present the parameter which will be varied, such as a control parameter like growth rate, stimulation, current, or environmental conditions. And on the y-axis, which is the vertical axis, we show the possible states of the system for a given parameter, such as the equilibrium values or periodic solutions or chaotic attractors. So we can put on the y-axis the equilibrium point, the amplitude of the oscillation or the steady state of the system. So we put on the x-axis the parameter being varied and the y-axis the parameter being monitored. And as the parameter changes, the diagram may show branches, splitting or merging, and this will indicate the different transition in the system behavior. And these transitions and changes are called bifurcations. And often we use solid lines to represent the stable states and dashed lines to represent the unstable states. And in some cases, stable and unstable regions may be color-coded or marked differently, but commonly we use solid and dashed lines for stable and unstable states respectively. And for the critical points where the system behaves differently or undergoes qualitative changes, we call it bifurcation points. So now the situation is simple. For the bifurcation diagram, we have x-axis, y-axis, and a certain curve. X-axis, the parameter being varied, the y-axis, the parameter being monitored. Inside the diagram, we have solid and dashed lines. Let's assume it's stable like that and unstable like that. So at this point, which we'll consider it a critical point or bifurcation point, the system changes from stable to unstable, from being stable like here to unstable, or unstable like here to be stable. And this also reflects the meaning of the branching behavior. So, or we may get a bifurcation like that. We have a system like that stable, but in this region, it's unstable. So at this point, which looks like a fork, we have a bifurcation point, which converts the system from being stable to unstable, if you are looking here to here. So now we know that nonlinear differential equations cannot be solved analytically, and we can depend on the numerical solutions or qualitative approaches, including the bifurcation diagram. And for the bifurcation diagram, we know that it's like a diagram, x and y axis, x the parameter being varied, y the parameter being monitored, and inside the diagram we need to find the critical point or the bifurcation points. There are many types of the bifurcations in bioengineering, such as a saddle node or transcritical or pitchfork or half bifurcation, and we are going to cover one by one and understand the meaning behind each of them and the graphical presentation of that system. So the first is called saddle node bifurcation, and here the bifurcation occurs when we have two equilibrium points. One is stable and one is unstable. Both of them collide at the bifurcation or the critical point and annihilate each other. As the parameter, it changes whether increasing or decreasing. And you can find that with systems like a critical threshold is closed or the system transitions from having two fixed points to none. And let's assume that we have the parameters that we need to change or vary and the output. And we have two equilibrium points. One is here and another is here. We mentioned by changing the parameters that we need to vary and monitor, there is something like that, and for the other which is unstable like that. And at a certain bifurcation value, they will collide together and annihilate each other at that bifurcation or critical point. This behavior can be represented mathematically using this canonical equation, dx by dt equals r plus x squared. And if you look here, it's nonlinear differential equation because we have a power of 2. We now have x, which is a state variable. It can be concentration, population size, and so on. And r is a parameter controlling the system behavior. And we need to change this parameter and understand the behavior of the system. So the parameter we are going to vary is r, and the parameter we are going to monitor is x. If you plotted the system, you will find that by changing the parameter, let's assume from -1 to 1, the parameter is r here in the x-axis, you will find that we have two curves because we have x squared. So we'll get 2 points. Let's assume here a starting point and here. And simply, we are going to plot r versus x. These two curves decreases as the parameter r increases. And both of them will increase is stable, and this is the unstable, until they reach a certain point, which is 0. And here, they will collide together and annihilate each other. So simply, you can say that at this region, we have a stable system. And we need to focus that our R value is below 0, which is the collision point or the bifurcation point. So once you have a system like that, you will find the critical point or the collision point. This will be your bifurcation point. And you will check which is stable or which is unstable. And if there's a region of the stability, then your X values should be below 0 and your R value should be below 0. At this region, you will get a stable behavior. But if you switch it to the other area where X is above 0, this will be the unstable area or your unstable curve. So based on that qualitative approach and the behavior of the system, you can decide that it's a saddle node bifurcation, and at a certain point it will collide together. So I know now what are the limits of the system. So again, the system behaves differently. R below 0, which is the collision point or the bifurcation point. There are two equilibrium points exist. One is stable and one is unstable. And at R equals 0, the two equilibrium points collide and disappear. Again, it's a bifurcation point. And here, the system undergoes a qualitative change. And for R above 0, no equilibrium point exists. And the system cannot sustain any steady state and may diverge or collapse. So you don't have at this point or at the system or at this equation, you don't have to increase the value of R. So now the situation is simple. You have an equation, and if you plotted it, you got something like that. Two equilibrium points that at a certain point, they collide and annihilate each other. Then it's a saddle node bifurcation. And here you can observe that as you are increasing the R value, the stable and unstable equilibrium points move closer until they reach the bifurcation point, which is in our example R equals 0. The collision happens and no more equilibrium occur. After increasing R to be more than 0 or the bifurcation point, the system lacks any stability or steady state stability. It can diverge, it can extend, it can collapse or diverge. And you need to determine the bifurcation point, which in our case r equals 0. Now you understand the idea. Plot your system. You may observe two curves, one like here, one like here, and once they collide together, determine the bifurcation point. Let's assume here it's 2, so r equals 2. This is our bifurcation point, which is the critical threshold or the critical point for our system. Another type is called transcritical bifurcation, and here this bifurcation happens when two equilibrium points, we can call it fixed points, exchange stability as the parameter r changes, which means that one equilibrium point transitions from being stable to unstable, and the other equilibrium point transitions from being unstable to stable. So we have a changing in the state for these two equilibrium points. The change will happen at the bifurcation point. We can consider this canonical equation, dx by dt equals rx minus x squared, and it's nonlinear because we have here x squared. Again, x our state variable that we need to monitor, and r is the control parameter that we need to vary. This is a presentation of the transcritical bifurcation diagram. So when you plot the function r versus x, increasing both of them or changing both of them together, you will find here two curves or 2 points. Let's assume one is here and another is here. And by changing the value of r, the first point, which is here, moves from being stable until it reaches the bifurcation point, and it will switch or transit to unstable. And for the other point, it's unstable, and at the bifurcation point, it's stable. And it's called the transcritical bifurcation or the transcritical change. So this is our critical point or the bifurcation point. When you have r less than 0 and x is 0, you have a stable region or a stable curve. And if r is above 0 and x equals r, will be this region. So we have two stability points or two stability regions. And also we have two instability regions here and here. So you can simply analyze your system behavior. For r below 0, the system has two equilibrium points, one at x equals 0 and one at x equals r. One is stable and one is unstable. At r equals 0, the two curves or the two equilibrium points collide. And at the stepping point or bifurcation point, the two equilibrium points transit from stable to unstable and unstable to stable. So x equals 0 moves from being stable to unstable, and the other one moves from unstable to stable. And the key feature of this transcritical bifurcation, as we mentioned, is the exchange of stability between two equilibrium points as r passes through 0, moving from stable to unstable and from unstable to stable. Again, based on your plotting of the system that you have, you can observe that if it's saddle point or transcritical or any type of the bifurcation, and you need to determine the bifurcation point or the critical point, and which is a stable region and unstable region, and this will be your constraints of the system. A third type is called pitchfork bifurcation, and here when you have a stable equilibrium point and at the bifurcation point it becomes unstable, and two symmetric new stable equilibrium points exist. So you will have as a bifurcation point three things. The original one will be unstable, and the new symmetric points will be existing to be stable. Let's assume the canonical equation like here. dx by dt equals rx minus x cubic. X is a state variable and r is the control parameters that we need to vary to measure the x value. And this is a presentation of the bifurcation diagram for the system r versus x. Now you have a point which is stable here, represented by the solid line, and as r varies, it's still stable until it reaches a certain value here, which is the bifurcation or the critical point. We have three things as we mentioned. We have unstable behavior for the original equilibrium point as r increases after the bifurcation point, and the new symmetric points, one is here and the other symmetric is here, emerge and exist after the bifurcation or the critical point. So the behavior of the system are below zero, we have stable equilibrium point, and at the bifurcation point or the critical point, the system undergoes a qualitative change. The original becomes unstable, and two new stable equilibrium points symmetrically exist around that bifurcation point. And after R is above the bifurcation, we have three equilibrium points. One is unstable, which is the original, and two are stable equilibrium. So for the saddle point, we have two equilibrium from the beginning to the end, and at the end, no more equilibrium points because they will collide together. At the transcritical, we also have two equilibrium points that will change the stability of the bifurcation point. And for this one, the pitchfork bifurcation, we have one equilibrium point that will be converted to three equilibrium points. One is unstable and two are stable. So the feature of this system is the symmetry breaking. The system is symmetric around x equals 0 with one only single stable equilibrium point, and after that the symmetry is broken where two new stable equilibria emerge symmetrically around x equals 0 and the original one will be unstable. The last but not least bifurcation type is half bifurcation, and this occurs when a stable equilibrium point becomes unstable and a limit cycle emerges as a parameter R changes. And let's consider the Huxley model for the half bifurcation. Again, V is a membrane's potential, W is a recovery variable, I is external stimulation, and epsilon below 1 is the time scale separation between fast and slow variables. We have two equations, and let's assume that we are going to monitor I versus V. We need to change the I value, which is external stimulation or the control parameter, and we need to monitor the equilibrium membrane's potential V. So we are going to change the I value. So here, as you see, by increasing the parameter I, it increases with a specific, let's say a specific curve or a specific slope. But until a certain value or a certain point, it increases with a high slope, and then it may return to the original slope again. So there are different changes with very small changes for the parameter I. The other thing is the existence for the oscillation or periodic behavior. So if you plotted for a long period, you may find something like that, a period or a rhythmic behavior that exists after the critical point. So the behavior for the different values of R, which is here the I or the external stimulation, at the beginning the system has a single equilibrium point at V and W, where I is below the critical point and there are no oscillations, and we can say the system remains at rest. But at the critical point, the equilibrium point transitions from being stable to unstable, and the system begins to exhibit a periodic oscillation or a rhythmic behavior. And after the critical point of the input or the external stimulation, the equilibrium point is now unstable, and we can say a stable limit cycle exists, and this represents a sustained oscillation, and the amplitude of the oscillation grows as I increases further. So the triggering here is simple. You need to find the critical point, and this can be observed from the curve or the presentation of the bifurcation curve or diagram. How? You have a certain system, you have a parameter that you need to vary, let's call it R or I or whatever, and you have also the parameters that you need to monitor, let's say V or omega or whatever variables that you need to monitor, and we need to plot both of them together. You have the system, let's say a couple of equations, and you are increasing the value for R and solving numerically the system to find the corresponding value for V, W, or the parameters that you need to monitor, and you will get different points. Let's say R versus V. Put both of them together in a 2D bifurcation diagram. On the x-axis, the one you need to vary, and V on the y-axis, the one you need to monitor, and you will get for sure a certain curve here. Depending on the curve, you need to look carefully for the bifurcation or the critical point. Let's say it's here. So you need to observe now what is the behavior of your system. Did the system converge from stable to unstable with new two stable equilibrium points, or did you have initially two equilibrium points that both of them transited from stable to unstable and stable to stable, or did the two equilibrium points collide together, or what happened to your system? Based on understanding the behavior using a qualitative manner, you will determine what are your constraints. You will determine what is the limitation of the R value and what is the corresponding behavior of the system based on these constraints. After understanding that, you will determine that your system that you built or you are experimenting on, that, for example, R will be limited from -2 to 3. Other than that, the system will behave badly in a dramatic way. Now, we didn't use any analytical solutions, just the qualitative diagram, such as the bifurcation diagram. We observed the behavior of the system with the help of the numerical methods. Because when you are going to plot both of the variables together, you have a differential equation, dx, for example, by dt equals to something. But you need x, so you can solve this numerically using a software solver to get the corresponding v or the corresponding x and so on. So we switch the gears from focusing on analytical behavior to numerical and qualitative behavior. Let's move to another concept called chaos. And chaos refers to deterministic systems that exhibit extreme sensitivity to initial conditions, leading to unpredictable long-term behavior, even though the system follow exact rules. So simply we have a system that, based on the initial conditions, can exhibit chaotic behavior through the long-term behavior. So simply we have a system which is deterministic and can look like a random system, but it's not random. And we can say it's impossible to determine over the long term, or we can say unpredictable long-term behavior. So we have a nonlinear system, which can be a 3D or 2D, three-dimensional or second order with delay. And for the long term, it doesn't settle to a fixed point or periodic orbit. And more of that depend on the initial conditions. So simply you can get two different initial conditions, C1 and C2 for the conditions, and one of them can settle and another cannot for the long-term behavior. And it can lead to a chaotic behavior and unpredictable behavior based on that initial condition. From that, we can put a set of conditions for chaos in bioengineering models. First, nonlinearity, and it's required for complex interactions. Feedback loops, positive or negative loops, amplify small deviations. Time delay, delays in response can destabilize otherwise stable systems. Dimensionality, as mentioned, we need 3 variables or second order with delay. Sensitivity to initial conditions, small changes can grow rapidly over time. So the idea here is simple. Based on the initial condition, it can lead to a chaotic and unpredictable behavior over time. Although it's deterministic and follows the exact rule, we cannot predict the behavior for the long term as it's nonlinear. So we cannot get confused between chaos and random systems. Chaotic systems are deterministic, not random systems. However, they are sensitive to the initial conditions. There are many examples, including the neural activity in epilepsy, where EEG signals during seizures show chaotic oscillations, and the small fluctuations in ion channel activity can trigger large-scale irregular firing. Cardiac arrhythmias, where the heart rhythm disorders may arise from chaotic electrical wave propagation. Population dynamics and circadian rhythms under stress are another 2 examples. So there are many examples where you can find chaotic behavior. So let's start by simulating the chaotic neuron firing using the Lorentz systems. So neuron firing patterns can become chaotic under certain conditions, as mentioned, the initial conditions. For example, during seizures or in response to external noise, we can use Lorentz system to model chaotic EEG signals and neural dynamics. But originally, this system was used with fluid dynamics. In the system, we have 3 variables, x, y, and z, and these are the different equations for the 3 variables. The corresponding simulation output is a trajectory that never repeats and shows sensitive dependence on the initial conditions. The system itself spirals around 2 attractors without ever settling, and the trajectories appear to switch randomly between the two loops, though governed by the deterministic rules. So we can use this behavior to mimic the EEG signals during epileptic seizures or irregular neural firing under stress or external stimulation. And if you are going to simulate the behavior between X and Y, you can find something like that. We have two loops or two attractors, and the system behaves in a chaotic manner without settling down. And if you change the initial condition from 111, because we have three variables, to 0, 1, and 2, the behavior changes without also settling down. And if you notice, for the first one, we have a big gap here and a small gap here. And for the second one, the gaps are minimized. However, we have a chaotic behavior. Another example for the chaotic population dynamics using the Rössler model. So in that model, small changes in the population levels or environmental conditions can lead to chaotic fluctuations. The Rössler system is a simplified model that accepts chaos with only one nonlinear term. We have three variables, x, y, and z. As mentioned, one of the rules, we need three variables or a second order with a time delay. And here we have a nonlinear term, z multiplied by x. Regarding the simulation output for that chaotic population dynamic system, it's a chaotic time series showing irregular oscillations in population levels. And unlike periodic oscillations, the pattern doesn't repeat exactly. So small changes in the environmental conditions can cause unpredictable long-term behavior. So if you are going to plot x versus y, you can find based on these initial conditions and parameters that the system has a chaotic behavior observed here, and the pattern cannot be predicted. And if you change the initial conditions from 1, 1, 1 and to 0, 1, and 2, the chaotic behavior changes. However, in both of them, it's unpredictable over the long-term behavior. Here's a third example for the forced van der Pol oscillators that can be used for the cardiac arrhythmias modeling. So we can use that model or this equation to model the phenomena of cardiac arrhythmias like atrial fibrillation, where it shows a regular, sometimes a chaotic electrical wave propagation. So the output is also a time series of membrane potential or cardiac voltage that becomes irregular and non-repeating under certain forcing conditions. So if you have low forcing amplitude, you can get regular restoration, which mimics the normal heartbeat. And for intermediate values, you can get period doubling leading to chaotic ECG-like patterns, where the trajectory no longer settles into a stable cycle. Instead, it wanders unpredictably. So if you are going to plot that, you can find during the time, you can get unpredictable oscillations. And if you change the conditions from 0 and 1 to 2.5 and 5, the oscillations behave unpredictably also. One more example for the chaotic behavior, we have the Marsh-Rose model, which is widely used to simulate the behavior in neurons. We have a nonlinear equation here and here, and we have three variables, x, y, and z, where x is the membrane's potential, y is the fast recovery variable, and z is a slow adaptation current, and the external force or external input is I. You can also get more information from this study about that model, but in all cases, if you are going to simulate the output, which is the voltage versus the time, it will show regular spiking, bursting, and chaotic transitions as parameters change. So if you have low input current I, the neuron stays at rest, and for moderate I, you will get regular spiking, which can be considered healthy behavior, and for high I, you will get chaotic transitions in your output. So as you see here, with these initial conditions and these inputs, we have 0.5 as an external input and initial conditions 1, 1, and 1, you will get that behavior, which is a membrane's potential. But if you change it, the I value to -5, you may get that value, or you may get one only spike, or you may get a chaotic behavior. So we can consider this also a chaotic behavior. By all means, we can conclude that the chaotic behavior or chaos in the systems that we are modeling requires set of conditions, such as being nonlinear, containing three variables or second order with a delay, and these chaotic systems or these systems during modeling can behave unpredictably over long time and impossible to predict. However, they are following the same exact rules and deterministic, but we can say that they are very sensitive to the initial conditions or small changes in the parameters. So in this lecture, we covered the definition of nonlinear differential equations, and we compared between nonlinear and linear differential equations. We moved then to two major concepts. One is called bifurcation and the other is called chaos. Bifurcation means that the system can get qualitative changes during varying a parameter or more, and we presented different types of bifurcations such as half bifurcation and saddle node bifurcation. And from that concept, we declared that nonlinearity systems cannot be solved analytically, but using numerical and qualitative approaches, we can solve or we can find the output or the corresponding output from these systems. And based on the bifurcation types and the definition of bifurcations, we can plot our system and know the critical points or the bifurcation points, and based on that, we can put limits or constraints to our system to work in this area. Then we switch the gears to chaotic systems and chaos, and we emphasize that there is a difference between random systems and chaotic systems. Chaotic systems are systems that are deterministic and follow exact rules. However, based on the different initial conditions, they may look random, but they are not random, and they are unpredictable, which are impossible to determine over long time. And we presented multiple examples to show the effect of the different initial conditions.